house of Mighty Rams, it's Ms. Hightower. I am coming to you guys live from my house, and I'm going to demonstrate to you guys the Pineapple Enzymes Lab. So we are going to be having a lot of fun as we investigate the role of pineapple enzymes on gelatin, which is a protein found in jello, okay? So this is what we're going to need today. We are going to need 2 types of pineapple. Bam! We got some pineapple chunks and canned pineapples, okay? So this, it's important to note that canned pineapples have been boiled. So keep that in the back of your mind. We're also gonna need, bam, fresh pineapple, okay? And then we're going to need some jello. I bought some orange jello, okay? And then you're gonna need a pot, access to hot water, cold water to make the gelatin firm up in the jello, and then some labeled condiment cups. So when I break you guys up into small groups, you're gonna have one that says boiled pineapple, okay, that's the canned one, and then you're gonna have a condiment cup for the fresh pineapple inside of the jello. So I want you to start putting your thinking brains on, and I want you to think, before we set up this experiment, on your hypothesis section of your note catcher, I want you to pause, stop and jot for a moment, and I want you to think about what will happen to the jello from the boiled pineapple, and what do you think is going to happen to the jello that has the fresh pineapple piece in it. So I want you to pause, stop and jot for a moment, and I want you to predict using the sentence stem displayed on the screen. All right, y'all, we made it to my kitchen, and now it's time to prepare the jello. So I have 2 boxes of this jello mixture, and I'm just going to follow the package instructions. So it says to add 1 cup of boiling water to package contents. So I just dumped both. Well, I got to finish dumping the second package of jello. There we go. Remember, this has all of the protein needed to make the jello firm. Okay, so the instructions say, let's bring it back up, to add 1 cup of boiling water to the contents. So that's exactly what we're going to do, and stir until dissolved. I've doubled the recipe, so I have 2 cups here. That's boiling. Perfect, perfect, perfect. So that's what we're going to do here. Oh, yeah. And we're just going to, oop, it just got steamy in here. So we're just going to stir just a little bit, okay, just to get this jello, the gelatin dissolved. And then I will add hot water in just, cold water in just a moment. It'll be 2 since I added 2 packages of jello. Oh, that is an ice ring there. That's how cold that is. Okay, cool. All right, stir, stir, stir. And now I have to go ahead and get the labeled condiment cups. Okay, to prevent cross-contamination, I'm going to have a separate fork for the pineapple that's boiled and for the fresh pineapple. So I'm going to also, to get this jello out of the bowl, I'm going to use just an 1/8 cup measurer, and I want to pour just a little bit in each of the labeled cups. Remember, they're labeled, one is boiled and the other is fresh. Okay, there we go. I think I put a little bit more, but not too much because we have to save some room for the pineapple chunks. All right, that's about even. So this is going to be the fresh pineapple. It's going to go straight in just like that. And then I'm going to use the new fork for the boiled or canned pineapple chunk. And it's gonna go just like that. Perfect. All right, before I put both in the refrigerator, now that I have both cups set, I just want you guys to make your day 0 observations. So here's the fresh pineapple inside of the jello. What do you see? What do you notice? And then here is the boiled pineapple in the jello. What do you see? What do you notice? Alrighty, fresh and the boiled. What do you see so far before I refrigerate? What do you notice? Go ahead and stop and jot and make those day 0 observations now. So now we have our 2 molds. I'm going to go ahead and make the other ones for the rest of your groups. I'm going to refrigerate them overnight, and then I'm going to bring them to class tomorrow so we can observe the effects. Which jello cup will get firm? Which one will not get firm? Or will both get firm? Will both not get firm? We will find out tomorrow. All right, Rams, now that I have officially demonstrated day 0 of the lab, all of the prep so you don't have to, it's time in your small groups to make your next day observations and notate them in your note catcher that your teacher has passed out to you. And notice, which jello cup has remained, or cups have remained solid or have solidified, which jello cups are liquefied. Make those notations below in your note catcher, and then it's time in your small group to turn and talk. Why? What happened? And why do you think these changes happened? Remember, tie all of your answers back to what we've learned so far about biomolecules, specifically enzymes, and then notate enzymes are proteins that help speed up chemical reactions such as digestion. So take all of that background knowledge that I or your other biology teachers have given you, and now it's time to apply and critically think. Why have these changes that you see? Why are they there? Why has this happened? Okay? It's time to use your critical thinking and your scientific reasoning to justify and explain this phenomena. All right, you guys. Happy thinking.